Alright, question 9 asks you to explain the steps in purifying a protein using a his tag. Start with the procedure for adding a his tag to your protein. Sorry, I gotta pull my pen up. Alright, so the steps here. So first of all, we need to get the his tag on our protein. Now, how do we do this? How do we actually attach the sequence of 6 to 8 histidines onto our protein? Well, we do it at a genetic level. So we basically, step 1 is we want to make recombinant DNA. Where your gene, YG, is fused with the his tag DNA. So basically, commonly this is done by amplifying your gene and inserting it into a plasmid right after or right before several codons coding for a his tag. That way, the 6 histidines are now going to be expressed as a part of your protein, as one, because that DNA will get converted into one mRNA molecule, and your gene sequence is now part of a gene sequence also containing these 6 histidines. All right, so once we have that DNA molecule made, all right, the second step is we have to transform our organism with the DNA. So basically, we have to get that foreign DNA, your gene, into the host organism along with its his tag. All right. Once we've done this, we can then have the organism express your protein. So we have to allow the organism to make it, so express your gene. All right. Then we start our actual protein purification. Now, when we do protein purification, our first step is always we have to, or almost always, we have to break open the cells. So we have to lyse them. All right. There's lots of different methods you can use here depending on which organism we're talking about. Now, once we've lysed them, now we have our protein mixture. Right. And so what we're going to do is we're going to bind our protein to the nickel column, or to the column beads. All right. Once our protein is bound, we can wash off any unbound proteins. All right. This is one of the aspects of protein column chromatography that really helps us when we're purifying any protein with any type of column, aside from gel filtration. That's a little bit different. But generally what we do is we bind our protein to the column and wash off anything that doesn't bind. In this case, most proteins do not bind to a nickel column because they don't have this sequence of 6 histidines. All right, now once we've done that, once we've washed off all these unbound proteins, we need to elute our protein. Now, for eluting a protein off a nickel column or with a his tag, I would want you to be a little more specific about how you can do this. And the ways you can do this are by adding in increasing amounts of a compound called imidazole. Or secondly, we can get our protein to release by protonating the histidines of the his tag, because if you recall, the lone pairs of electrons on histidine are necessary to bind to the nickel, the metal ions. So how can we protonate it? Well, by decreasing the pH. Those are the 2 ways we elute our protein off of a nickel column. And that is it for this problem.